Sorry, we got cut off last time. My program that I'm using only allows me 10 minutes of video. I promise we're not going to go much longer than that. Um, but we want to write these instructions down. So the ribosome builds the protein and it sends it through the ER. The ER is then going to basically kind of be a transport vesicle where it transports it to the Golgi apparatus. The Golgi apparatus will basically package and sort the proteins, decide like what needs to go where, and then it's going to ship it to the rest of the body, or not body, but cell, like wherever it needs to go within the cell. All right, so a big way to tell the difference between an animal cell versus a plant cell is one, they're usually circular in nature, and then some of the things that they have in it. So, for instance, you'll look for centrioles. Centrioles are going to be these guys right here. Okay, so they're circular in nature. You'll look for centrioles, but notice again, we have the endoplasmic reticulum right outside of the nucleus. So we have the DNA. And so when we think about proteins, this is where the instructions come from. The DNA is copied in a form called RNA. Don't have to worry about that yet, but then it's taken to the endoplasmic reticulum where the ribosomes are, and a protein's made. That protein is then shipped in a vesicle to the Golgi apparatus where it's packaged and sorted and then shipped throughout the rest of the cell. Okay, so an example, if you were to look at one under the scope, we know this is a plant cell or animal cell because it's circular in nature. Okay, it's actually cheek cells. We could actually do this. It's a really simple thing to do. You just use a toothpick and then you swab it onto a slide and you'd be able to see this. You'd have to put some stain on it. That's why it's blue. But right here is the nucleus, which is kind of cool to look at. Plant cells are going to be different, and we're going to look for different things. So the three things, or the three organelles that plant cells have that animals don't, is a cell wall that's going to help provide additional support, so structure and support with the cell wall. Chloroplasts, they contain the green pigment that allows them to go through photosynthesis, so where we turn light energy into food. And then the central vacuole is another indicator. So when we look at a plant cell, so here, this is Eladia. You used to be able to buy this, but then it became, if it infests our waters, it overgrows and it becomes this invasive species we can't get rid of. So you can't actually buy this at stores anymore. But this right here, you can tell here is the cell wall. You can see like that thick layer in between. And then all these green little dots are all the chloroplasts within each of the cells. Now, when looking at a plant cell, things to kind of recognize is that the cell wall is on the outside of the cell and the cell membrane is on the inside. Okay, kind of remember that. A big way to tell the difference between a chloroplast, which is this one right here, goes through photosynthesis, and maybe a mitochondria is, again, chloroplasts have these like little spree packaging in them. And then the mitochondria in decent pictures, this is not the best picture, but mitochondria has this folds on the inside. Remember to increase surface area so that more reactions can take place. But another big indicator is this large central vacuole, and that's how you would know it's a plant cell. Last but not least, and we're actually going to spend a couple days on this, and I think you'll have fun because we're going to play with bubbles, is the cell membrane. A cell membrane is semi-permeable, meaning it allows some things in and then some things can't go in, which is important. We don't want toxins inside of our cells. So it regulates what comes in and what doesn't come in. And it's composed of something called phospholipids, and it's actually a bilayer. So a phospholipid bilayer, meaning that it binds two. Okay, so it has two layers. So this right here is a phospholipid. It has a hydrophilic end, so that means it's water loving. Hydro means water, so it's water loving, and it has a hydrophobic tail, so it's water hating. So it associates in that certain way, which is really good for our cells, and we'll talk about why that's good. But it also has proteins embedded through it. We're going to spend more time on this, but that's pretty much it for our lecture today. And hopefully you got everything written down. The next thing you're going to do is your cell organelle concept map or your cell organelle word map. And there's a video that'll show you how to do that. So that is the end of our lecture.